Good day, grade 12 learners, and welcome to this video where we will be looking at section A questions taken from previous exam question papers to prepare you for your paper 2 of your NSC final examination. Please ensure that when you are preparing for paper 2, you are studying the following topics: DNA, the code of life, meiosis, genetics and inheritance, as well as evolution. The first question is taken from a question 1.1 that states various options are provided as possible answers to the following questions. Choose the answer and write only the letter A to D next to the question numbers in your answer book. For example, 1.1.10, and if you choose the letter D, then D is going to be your answer. Please ensure that you only have one correct letter written in your answer book. You can't have more than one correct letter or else it will not be able to be marked. So 1.1.1, the various factors controlling the different characteristics are separate entities, not influencing each other in any way and sorting themselves out independently during gamete formation. This refers to Mendel's principle of segregation, Mendel's principle of independent assortment, Lamarck's law of the inheritance of acquired characteristics, or chance fertilization. You can see the most correct one here is going to be B. Therefore, the answer is B, Mendel's principle of independent assortment. 1.1.2, multiple alleles exist when A. One allele controls the inheritance of a gene, B. Two alleles control the inheritance of a gene, C. Alleles are inherited from two parents, or D. More than two alleles control the inheritance of a gene. Multiple alleles exist when more than two alleles control the inheritance of a gene. Therefore, D is correct. 1.1.3, how many nitrogenous bases are found in a protein made up of 66 amino acids? Remember that one amino acid is going to comprise of 3 nitrogenous bases. If you are going to have 66 amino acids, you are going to multiply 66 amino acids by 3. You type it in the calculator, the answer will be 198. Therefore, A is the correct answer and the rest of them are all incorrect. 1.1.4, a structural difference between nucleic acids is in DNA is the double-stranded molecule. Yes, it is. RNA single-stranded. Yes, that's correct. DNA contains nitrogenous base uracil. No. RNA contains nitrogenous base thymine. No, it does not. DNA is a straight-stranded structure. No. RNA is a helical structure. No. DNA is a shorter molecule and RNA is a longer molecule. No. Therefore, the correct option is A. 1.1.5, the diagram below shows a phase in cell division. So this is the phase they're referring to. The phase in the diagram above is the prophase 1, telophase 2, telophase 1 or just telophase. You can see that you have individual chromosomes here. Therefore, it is going to be telophase 2 and the answer is B. The next questions 1.1.6 and 1.1.7 are based on DNA profiling below. So 1.1.6, the parents of individual B are. So we are looking at individual B and we're going to compare individual B to all of the other DNA profiles A, C, D, E and F so you can possibly get our correct answer. So we match B with A, we have 1, 2, 3, 4. There are 4 matching bars. Then we match B with C. From the top, we have 1. We only had 1. Then we go to D. There's 1. There's 2 and there's 3. There's 3 matching bars for D. We go to E. There's 1. There's 2. There's 3. There's 4. There are 4 matching bars with E. Then with F, nothing's matching, nothing's matching, nothing's matching. There's only one matching bar with F. Therefore, the parents of individuals B. So we have 4 matching bars for A, we have 4 matching bars for E. Both of it is going to give us 4. Therefore, if we go to A and E, C is going to be the correct answer. 1.1.7, which one of the following options is not used for DNA profiling? Matching tissues for organ transplant? Yes, that is used. Diagnosis of inherited disorders? Yes. Developing cures for inherited disorders? No, we don't develop cures using DNA profile. Biological evidence in criminal cases? Yes, therefore C is the correct answer. 1.1.8, the diploid chromosomes composition in a normal human male. 44 autosomes and XX chromosomes? No, because X and X will be in a female, and you want to get to your 46, so that will give you 46 chromosomes in a female. 22 autosomes and Y chromosome, it's 22 autosomes, it's 22 pairs of autosomes. It's not 22 autosomes, there are 44 autosomes, therefore that's incorrect. 22 autosomes and X and Y? No, because it must be 22 pairs. 44 autosomes and X and Y? Yes, because that is showing a male, therefore D is correct. 1.1.9, a mutation can have the following effects: code for the same amino acid, change the protein formed, cause diseases in an organism, not be expressed in the organism's phenotype, increases chances of survival. Which one of the above combination is only due to harmful mutations? Harmful mutations, they code for the same amino acid? No. Change the protein form? No. Cause diseases? Yes. Not be expressed in the organism's phenotype? No. Increased chance of survival? No. Therefore, 3 is the only correct one, and C is going to be the correct answer. The next question is a 1.1.6 that says the diagrams below represent the 4 limbs of 4 animals that all contain similar bones. Study the diagrams and the statements below. So we have the human, the cat, the whale, and the bat, and we can see that what the diagrams are looking like. The structures are homologous. The animals do not share a common ancestor. The 4 limbs of all these animals have the same function. The structures arose due to modification by descent. Which statements above apply to the diagrams? So the structures are homologous. Yes, they are. The animals do not share a common ancestor. They do, so that's wrong. The 4 limbs of all these animals have the same function. No, they don't all have the same function. Because all the bat, the whale, the cat, the human, these limbs don't have the exact same function. The structures arose due to modification by descent. Yes, it did. Therefore, we chose number 1 and number 4, which is therefore going to be C, the correct answer. The next questions, 1.1.8 and 1.1.9 are based on the diagram below. So we have our phylogenetic tree along with a little timeline here in millions of years ago. And we see it is moving from 0 and it's going down all the way to 6. Approximately how much longer do pan troglodytes exist compared to its closest relative? So we locate there, we're looking at pan troglodytes. How much longer did it exist compared to its closest relative? And here we have its closest relative, Australopithecus afarensis. If we look at this chimpanzee here, it located when? Until 5 million years ago. So we subtract this 5 from 1.4, which will give us 3.6. And therefore the answer is 3.6, which is going to be B. So we take this 5 minus that 1.4 for Australopithecus afarensis is going to give us 3.6 million years ago. Then 1.1.9, how many genera are represented in the diagram? Genera, remember the first name is the genus's name referring to the genera. So we have Homo, we have Australopithecus, we have Paranthropus, and we have Pan. So we have 1, 2, 3, 4. And therefore the correct answer would be C. The next question, we are looking at our terminology questions for 1.2, which states give the biological term for each of the following descriptions. Write only the term next to the question numbers 1.2.1 to 1.2.9 in the answer book. So question 1.2.1, the representation of the number, shape and arrangement of chromosomes in the nucleus of a somatic cell is referred to as the karyotype. The location of extranuclear DNA in plant cells only is the chloroplast. The point of contact as non-sister chromatids overlap each other in crossing over is referred to as chiasmata or chiasma. A blood type that has two recessive alleles is going to be blood type O. The position of a gene in a chromosome is referred to as the locus. A strand of DNA where nucleotides attach to build a new DNA strand is regarded as the template strand. A condition characterized by an extra chromosome at position 21 is Down syndrome. The type of bonds that hold nitrogenous bases in a DNA molecule is referred to as weak hydrogen bonds. A condition of the cell where there is only one set of chromosomes is going to be haploid. The allele that does not influence the phenotype in the heterozygous condition is going to be referred to as the recessive allele. Because remember, in the heterozygous condition, it's the dominant allele that's going to be expressed in the phenotype. The bond that forms between amino acid molecules in a protein is a peptide bond. A portion of a chromosome that codes for a particular characteristic is referred to as a gene. The biotechnological process that produces a genetically identical organism is referred to as cloning. The type of variation where there is a range of intermediate phenotypes is referred to as continuous variation. The study of the past and present day distribution of living organisms across the continents is referred to as biogeography. The scientists argue that acquired characteristics obtained by parents could be passed on to their offspring is going to be Lamarck. The next question looks at 1.3. We will indicate whether each of the following descriptions in column 1 applies to A only, B only, both A and B, or none. 1.3.1. The type of variation that exists in blood groups, codominance and discontinuous variation. That exists in blood groups, codominance and discontinuous variation. Does it occur in blood groups? No, only B. Discontinuous variation is going to occur in blood groups. 1.3.2. Cytokinesis occurs twice in meiosis or mitosis, only in meiosis, because meiosis has telophase 1 and telophase 2, therefore A only. 1.3.3. The absence of blood clotting disorders, hemophilia or color blindness, only hemophilia, therefore the answer is A only. The next table with 1.3.1, the characteristic of an African ape skull, prognathous, less pronounced brow ridges of an African ape, prognathous is the only one, therefore A only. 1.3.2, characteristics of blood group inheritance, multiple alleles or codominance, blood group inheritance, multiple alleles, yes, codominance, yes. Because one of the blood groups are going to have codominance, blood type AB has codominance, therefore both A and B are correct. 1.3.3, in a cross between a bull that is heterozygous for long eyelashes and a brown coat color and a cow that has short eyelashes and white coat color, the gametes of the cow will be. If you look at A and if you look at B, they are giving you gametes there. And the actual alleles was not even given to you. Therefore, none of these can be correct and the answer is none. The next question is question 1.4 that says growth hormone is important in promoting skeletal and muscular growth. It can be produced for people who do not secrete or undersecrete this hormone using recombinant DNA technology. So you have the human growth hormone production using recombinant DNA technology. You have steps 1, human cell. Then you have the bacterial cell with bacterial chromosome. You have structure X that was removed. Then you have step 3 with the sticky ends. You have the gene for human growth hormone taken. You're forming your recombinant DNA. The gene for the human growth hormone then got inserted, and then in step 5, you have your bacterial cell containing gene for your human growth hormone. 1.4.1 says name the biotechnological process. Name the biotechnological process shown in the diagram above. This biotechnological process is obviously going to be referred to as genetic engineering. 1.4.2 says identify structure X found in a bacterial cell which is used in the above process. Structure X in the bacterial cell that you are removing here is referred to as the plasmid. 1.4.3 says name the organic catalyst used to cut structure X. So you are cutting structure X. What is the catalyst that you are going to use to cut structure X? It's referred to as an enzyme. 1.4.4 give one benefit of using the growth hormone by sports athletes. What is the growth hormone benefit going to be in sports athletes? It's going to be promoting skeletal and muscular growth. The process named in 1.4.1 for diabetes, so the process named in 1.4.1 was genetic engineering. What is the benefit of genetic engineering for diabetic patients? It's going to allow them to have insulin, so it produces insulin. 1.4.5 state 2 characteristics of bacteria that make them suitable to be used in the above process. So what does bacteria have that allows them to be used suitably in the above process? Firstly, they reproduce very rapidly. Secondly, they reproduce asexually or by mitosis. Bacteria exist everywhere. They are simple organisms. And DNA is in the form of a plasmid. 1.4.6 describe one observable function of the sticky ends as illustrated in the process above. So the sticky ends that you have here, what is one observable function of them? If you move on to step 4, you can see that the sticky ends is going to have the gene for human growth hormone inserted into it. Therefore, 1.4.6 attaches the gene for the human growth hormone to structure X or to the plasmid. So 1.4.6 it attaches the gene for human growth hormone to structure X or to the plasmid. Because you can see here, this is going to be your structure X, and the gene for human growth hormone is inserted to the plasmid because of that sticky ends that you have. The next question, 1.5, says the diagram below shows two models used to explain the theory of evolution. So you have model 1 pointing to this line, and then you have model 2 pointing to the second thick line that is illustrated. 1.5.1, identify the evolutionary theory in model 2. So model 2 is going to be that one there. So therefore it's going to be referred to as punctuated equilibrium. 1.5.2 says name the two scientists who proposed the theory in model 2. So that was going to be Niels Ehlers as well as Stephen Gould. 1.5.3, which evolutionary model, 1 or 2, is showing small gradual changes in organisms from one generation to the next. Small gradual changes from one generation to the next is going to be model 1, because you can see you're looking at the straight line. If we chose model 2, that had to be rapid changes occurring. That's for punctuated equilibrium. Therefore, we can't choose model 2. Supported by the absence of transitional fossils is going to be model 2, because punctuated equilibrium did not have transitional fossils. Supported by scientists who propose a theory of evolution by natural selection, model 2 can't be natural selection because model 2 is punctuated equilibrium. Therefore, it has to be model 1. 1.5.4 state what happens respectively to species at points A and B in model 2. The species at points A and points B in model 2, what is happening? So if we have a look at point A, there's going to be no organisms do not evolve or change. But in point B, the organisms are changing, they are evolving rapidly. Therefore, the line is starting to spike up and it's starting to go up. 1.5.5, name the type of evolution where there's a change in characteristics of species over time. That is obviously going to be referred to as biological evolution. 1.4, the out of Africa hypothesis states that modern humans originated in Africa and they migrated to other continents. Some fossils of the genus Australopithecus have been discovered in South Africa. Firstly, give the name of the scientist who discovered Mrs. Blaze. The scientist was going to be Robert Broom. Then Littlefoot was going to be Ron Clark. 1.4.2 state the fossil site where Littlefoot was discovered. Littlefoot was discovered in Sterkfontein Caves. 1.4.3, the species name of the fossil Karabo was Australopithecus sediba. 1.4.4, name the genetic evidence that supports the out of Africa hypothesis. Because they are speaking about genetic evidence, remember genetic evidence is got to do with DNA, and the type of genetic evidence that helped to support the out of Africa hypothesis was the mtDNA, the mitochondrial DNA. That brings us to the end of this video where we revise questions based on section A in preparation for your paper 2 of your Life Sciences NSC final exam. Please ensure that you are studying the following topics for paper 2, DNA, the code of life, meiosis, genetics and inheritance, as well as evolution. Thank you very much for taking your time to watch this video.